about cellular respiration, or the reason that your seventh grade teacher taught you and then your ninth grade teacher allowed you to say the mitochondria is the powerhouse of the cell. If you say that to me from here on out, I will go, because I don't want to hear it out of your mouths anymore. From here on out, it is not specific enough. You are better than that. And you are going to give this little organelle all of its due, because it does a whole heck of a lot for you in terms of converting this stuff, the food you eat, and the oxygen you breathe in, so that your cells have the power and the energy that they need to carry out cellular function. Don't come at me with that mitochondria is the powerhouse of the cell anymore. Please and thank you. Here we go. So, first off, generals, I hope that this is a reaction that you can just write out right now. If it's not, this is going to need to be one of those ones that you just stick in your brain. Put it on a flashcard, do what it is that you need to do, but you need to know not just the reaction, but the complete balanced net reaction for cellular respiration. And that would be that it is one glucose molecule, one C6H12O6. Yes, you need to memorize the chemical formula for glucose, C6H12O6. Combine that with 6 oxygens, and it produces 6 water molecules, 6 carbon dioxide molecules, and that energy that we are talking about. Remember, the energy that we are talking about is not just power. It has a name. It is a nucleic acid derivative. It is adenosine triphosphate, better known as ATP, and you can think of it like cell cash. We're going to make a whole lot of cash throughout this process, and then we're going to spend it. You learned a whole lot about ATP in your ATP group activity, and here is where we need to use that information and that knowledge, as well as in the photosynthesis pathway information. Remember, this whole pathway and that of photosynthesis must follow the laws of thermodynamics. Those are just the laws of this planet. Specifically, we are only talking about the first law in this case. It is something you are familiar with. And it is that law of conservation of matter. That matter is not created, it is not destroyed, it is only transformed. We are not doing anything crazy in here. We're just simply taking one form of energy and we're turning it into something else. And we are not losing stuff along the way. This is a 4-part process, 3 of which you are going to be intimately familiar with and highly responsible for. One of which is I'm going to show it to you, and then you're going to go, okay, and then we'll move on. So we're going to go through those 4 parts. I don't know why I animated this slide like this. I was real excited. Like, really excited, guys. I'm sorry. All right, here are your 4 steps. You're going to start in the white. We're going to start on the very outside. Step number 1 is something called glycolysis. This is a slide that I would honestly print, white everything out, slip it in a sheet protector, and then practice filling it in. Okay? I will, if I'm feeling generous, provide one of those for you. Step number 2, we're going to move to the purple. You are going to process a molecule that we'll talk about called pyruvate. From pyruvate, we're going to move to step 3, better known as the citric acid cycle. All 3 of those parts together, the goal has just been to take one glucose molecule from the foods you eat and to break it down. The last step in the process, step number 4, is known as electron transport or chemiosmosis, and the goal in that portion is to actually make the ATP. It's a lot more intricate than just produces power. Yeah, but how? So here we go. 2 things that I need you to know before we can get started. These are going to be used now. This one is going to be used now and in photosynthesis. The next one that I'm going to tell you is only used here. These are enzymes. There are 2 enzymes in here. The function of this particular enzyme is to move one hydrogen molecule back and forth, creating various concentration gradients where we need them to be so that we can use other enzymes like pumps to make other things happen. This is the first one. You can see the similarity in the 2 structures. Take a second and see if you can't find the difference. I also highlighted it in the letter at the top. So hopefully at this point you have found that phosphate group hanging down off that hydroxide ion in the bottom right side. This is an enzyme. It's called nicotinamide adenine dinucleotide. It literally is just to move hydrogen ions from one place to the other. You're going to see it a lot. It is very abundant in both of these processes. It functions really similarly. Put this image in your brain. That's all this is doing. See that H? We're just moving an electron back and forth. Do we have it? No, we don't. No, we do have it? No, we don't. It's literally a never-ending game of ping pong. Seesaw is another great way to think of this. It will never end. The other molecule that I need you to know is this one called flavin adenine dinucleotide. It is a coenzyme. So it's not one on its own. It's a partner friend. You can see the complete balance reaction on the bottom. But when we put this FAD with 2 hydrogens and 2 electrons, we create a molecule called FADH2. Again, it's just an electron transport molecule. It's an enzyme. All we're doing is moving that electron back and forth. In this case, because of its structure, it moves 2 electrons at once. In the last one, NAD only moves 1 electron at a time. FAD is able to move 2 electrons at any one given time. And remember, sometimes we're going to say electron, and sometimes we're going to say hydrogen plus, really from a valence electron connection point, same thing. Okay, we're moving hydrogen. In this particular pathway, all of cellular respiration, we are in the mitochondria, but more specifically than that, we are inside the mitochondria in a space known as the inner mitochondrial membrane. So that's the kidney, like swirly, ribbony looking part on the inside. And we're inside of the inside. We're also going to be in a place called the mitochondrial matrix. So we're on the very inside. As you take a look at this structure, I hope you identify the 2 organelles, well, one's an organelle, one's not quite an organelle, that are unique to this particular organelle. That's right, it's an organelle within an organelle. Weird, right? If you have noticed those little dots on the inside are known as ribosomes. And then hopefully you also notice those circle things. That's DNA. Yes, your mitochondria carries its own little teeny tiny bit of DNA. We'll talk about why later. For now, I just want to make sure that you are always taking a look at a whole diagram and looking at every single piece that is in there. Again, we'll come back to this later. First step in the process, this crazy thing called glycolysis. Glycolysis is going to take the glucose molecule, one single glucose molecule, and it is going to rip it apart, and it's going to transform it into smaller molecules. The point of doing that is to create free energy. This actually doesn't take place in the mitochondria. It actually takes place outside the mitochondria, really close by, but just in the cytoplasm. There's no need for any oxygen. In this case, it's pretty impressive that you can go through part of the cellular respiration process and not have any need for oxygen. So we're going to grab that one glucose molecule. We're going to transform it into 2 pyruvates. And, oh look, there's our NADH. That is there. And we're going to start to make the first little bit of our money. So we're going to have 2 ATPs that we generate at this point. This is the point. This is all I need you to know about the process. So if you can visualize the top, It'd be great if you could write it out just in terms of C6H12O6 is what glucose is. Pyruvate, I want you to go through and figure out what that is. How many C's, how many H's, how many O's. That's all that's involved. Plus 2 NADH plus 2 ATP. If you just put that on a flashcard, that is sufficient. If you are a bit of a chemist and you're like, ooh, but I really like chemistry. Okay, there you go. If you're not a bit of a chemist and you're like, well, please stop. Don't worry, I will. That thing in the background that is gone now, holy bananas. Look at how many different enzymes are involved to take that 6-carbon structure and turn it into 2 3-carbon structures. That's really all I need you to know. So hopefully back here when you wrote out what pyruvate is, you recognize that there are 3 carbons in this bad boy. One that's designated on the end, one with a double bond to the oxygen, and then another one with a double bond to the oxygen. That is all I need you to know. We're going from a 6-carbon structure to a 3-carbon structure. That's it. That's all that you got to know. In fact, I'll even do you one better. If you could just know this, that's sufficient. That's really all that you got to know. Okay, glucose with some ATP, we got to spend a little bit of money here, turns into 2-triose phosphate. Then we're going to get that 2 NADs and those 2 ATPs, and we're going to turn it into the 2 pyruvates. Again, if you want to track and look at everything, go back in the slides. Heck, go to your textbook. Heck, just Google the word glycolysis, and you will find this diagram in 75 different forms out there, and then you can take a look and you can actually track and follow all of it if that is something that is of interest to you. It's a really beautiful organization of everything that happens. I just need you to know that it goes from glucose to 2-pyruvate. You get 2 ATP in the end, and you get 2 NADH. If it says reduced, that means there is an H. So it should be NADH, not H2, 2 NADH. Moving right along. We are headed to the link step in here. It's that purple step that you looked at a second ago. It can go by 2 different names. The problem here is that, yes, we have taken that 6-carbon structure and we've chunked it into 2 3-carbon structures. The problem is for your mitochondria, that's still not small enough. So we got to get it down even smaller. So out in the mitochondrial membrane, so we're caressing as we're moving from the outer, so the cytoplasm across that outer membrane, we are going to take that 3-carbon structure and we are going to turn it into this thing that you see called acetyl coenzyme A. If you love chemistry, this is what it looks like all the way drawn out. And you'll notice you can follow the 3 carbons that are in there. It's got a few more at this point. I just need you to know that we're going to take those 2 pyruvates, so those 2 3-carbon structures, and we got to turn it into this thing. At this point, we're going to have 2 NADHs. They're still along for the ride. We haven't done anything with them. We've got some 2 NADHs that are coming, and we've now made some carbon dioxide. And hopefully you know that carbon dioxide is nothing in cellular respiration but waste. It was on the product side. We just breathe it out. Every time you take a breath, you are losing carbon dioxide. The reason you're losing carbon dioxide from your body is because all of your cells have this byproduct of carbon dioxide at some point, and then it's a gas. So as you are breathing, it gets expelled from you. This is all I need you to know. I am not even going to show you the crazy bananasness that goes into that. If you are curious about it, knock yourself out. You have access to the internet. I don't need you to know anything other than we're going to turn pyruvate into acetyl-CoA. Okay? From here, we are going to move to a cycle that you need to know a little bit more about. I'll walk you through it. The point at this part of the process is to get the electrons that we need in the right place to create a concentration gradient that is useful for converting a DP into a TP. So all we're doing at this point is just moving stuff from point A to point B. From here, with that, once you have become acetyl-CoA, we can diffuse into the mitochondrial matrix, so that blue part in the middle of all of this stuff. At this point, we're going to need the oxygen. It is a necessary component to getting the whole thing to happen. And this is your general big picture reaction for everything. We're going to take just one of those acetyl-CoAs. Remember, there really are two. We are going to reduce it into 3 NADHs and 1 FADH2. Remember, that's really and truly all that we care about at this point, is just getting that to be produced. Also, while we're going through this whole process, we do get to make a little bit of cash here. So we're going to end up with 1 ATP molecule. Okay, and again, remember that for every glucose molecule, you make 2 pyruvates. Each one of those pyruvates turns into acetyl-CoA, and then each one of those breaks down. So really, you need to double all of the things that are happening in here, but we don't write it that way. We write it for a singular, and then you just have to remember that it really truly has happened twice. Okay? It's also known as the Krebs cycle. It was Johann Krebs that went through and he figured all of it out. Pretty sure that's his first name. He was an old dead white guy. Go through the whole cycle. You can see how everything works. Acetyl-CoA, we get rid of the coenzyme A. We're going to reuse that bad boy because it's a coenzyme, so it's reusable. We're gonna take that and we're gonna go back to a 6-carbon molecule. And if you're thinking like, what, why 6 to 3 to something else to now back to 6? It's all about diffusion. Certain things just cannot diffuse across the membranes. Remember, they're all phospholipid bound, and so you have to make them smaller, and there are no protein channels that will get this stuff through, so we got to make it small enough to get through. You can see the carbon dioxide that is given off right in the very first step as we come through the process. We're going to drop it almost straight away down to a 5-carbon molecule and get rid of our second carbon dioxide. So that's why there's 2 that come off of there. Carry along, skip, moving on, blah, blah, blah. And we are down to a 4-carbon molecule called oxaloacetyl acetate. Okay, yep, I know, big fancy word. Guess what? You don't need to know that. You just need to know this part. 1 acetyl-CoA, we get 3 NADHs, 1 FADH2, and 1 ATP. And you do it twice. That's it. I know my chemists that I'm not giving you the full Krebs cycle. If you love biochemistry, go take a biochemistry class that has something to do with cellular respiration, and you can learn the entire Krebs cycle in all of its molecular glory. I don't remember it at this point, so we're not going to do that. From here, we're headed to the last step in the process, better known as oxidative phosphorylation. In oxidative phosphorylation, we are going to get the energy, finally, necessary for organisms to function. We are doing this by creating an electrochemical gradient so that we can move things from point A to point B and then turn a crank. Just like all sorts of energy production in the real world, not that this isn't the real world, but things like wind power, solar power, hydroelectric power, you name it, geothermal power. At some point, you're turning a turbine. Guess what? We're going to turn a turbine here too. It's just an itty bitty teeny tiny molecular turbine. This time we are moving, we started on this cytoplasm, right? We went to the outer mitochondrial membrane, then we went into the blue matrix. Now we're going to come back across and we're going to go into the inner mitochondrial membrane. So we are in the ribbony part, right, of the bean-shaped thing that I showed you right here. So we're in the bean part in the middle here in that like ribbony looking part. Okay, so we do need oxygen. Again, at this point, without oxygen, this isn't going to happen. When you don't have oxygen, the last 2 parts, if you're asking yourself, like, well, what if? I'll get there. And the short answer is this doesn't happen. We'll talk more. Here's what you need to know at this point. We're going to take those 3 NADHs that we finally created in the last step, the 1 FADH2 and 1 ATP, and we're finally going to crank out the 34 ATP that are producible in this entire reaction and 6 water molecules. So we're finally going to get cranking all those pieces out of there. You'll notice up here we have a two-part process. The top part is known as the electron transport chain. The bottom part, and really just this little blue one, sorry, that one arrow is in the wrong spot. The two ovals are your electron transport chain. Your circle is chemiosmosis. Okay? So let me walk you through using this diagram. This is another really great diagram that you might want to, this one you could probably just draw by hand, but you could also print off this slide, white these pieces out, pop it in a sheet protector, and just write it down over and over and over again until it sticks in your brain. So here's what happens. First off, orient yourself. Inner membrane space, so this is the, like, towards the outside. Matrix out here on the inside, okay? Remember, we have made a ton of NADH, that's the top purple one, and we've made a ton of FADH2, that's the top green one. We're gonna rip the hydrogen off of those two bad boys. When we rip the hydrogen off of those two bad boys, those two coenzymes, thank you very much, we are gonna harvest their electrons. And when we harvest their electrons, they're gonna go into this pump, we'll call it, okay? In this pump, what it does is it actually takes the electron and it doesn't pump it through, it pumps it across. So it's going to send it over to its friend next to it, the other pump. I don't know what its name is. You don't need to know what its name is. Unless you get really into oxidative phosphorylation and the electron transport chain, just know that this is what it kind of looks like. It's like a really long slide with multiple parts, and you can like jump different parts. It's like the best, I don't know, Busch Gardens ride ever. And so you're going to move, and then you're going to hop to this guy. When you are at this one, that H that was ripped off back here in the NAD and the FADs, we're going to pump it from the inside in the matrix. We're going to pump it from the inside to the outside. We need to do that because we need to create a concentration gradient differential. And we need it to exist no longer up in the blue matrixy part. I need to get that down to the other side. Hold that thought up in terms of why. From here, we're going to continue down on our amazing, I don't know what those things are called, but our amazing ride that we are taking here. The electron ends up down here. The electron's kind of cool. It does its own thing. It is high energy. It gets to do whatever it wants. Remember I told you that oxygen was necessary? Well, here's where it's necessary. Because we're going to steal one of these H's. We're going to take that oxygen and we're going to turn it into water. We do that, we're going to use that one electron to do that. We only needed the electron and the hydrogen ion to create the difference. We got to now get rid of some of those. That's a little much. We do that in the form of water, so we get rid of that. We now have a tremendous number of hydrogen ions that are in the intermembrane space. Those get pumped. Remember I told you I would talk about pumps eventually and active transport. Those get actively transported via ATP synthase. This was also one of the models at the very end of your group activity on membrane structure and function. We're going to pump some of those hydrogen ions back across through this enzyme called ATP synthase. When we pump hydrogen ions across the ATP synthase active transport pump enzyme, it turns a little turbine up here at the top. That turbine, when it turns, takes one ADP adenine diphosphate and it reattaches a phosphate group to it, churning it, converting it into adenine triphosphate. So now we have that one back at the end. Remember, the whole point of making ATP is so we can rip those phosphoruses off one at a time. When we do that, it releases a tremendous amount of energy. Releasing a tremendous amount of energy allows us to function, and it allows all organisms to function, plants included. And then this process just continues on in perpetuity forever and ever and ever and ever until one day you die or the plant dies. And then it still happens a little bit more, and then eventually it just gives up life, and then it is done. Okay? I like this diagram because it kind of just does it as a little GIF, does it in real time. You can see those electrons, the water molecule, pump, pump, pump. Oh, ATP, 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 ATP. This one says 32, I wrote 34. It is a wide variety of numbers and it can fluctuate every time this process happens. Okay, just gonna take it in, sit with this diagram for a minute, make sure you fully understand where everything is going and how it's moving. Don't watch it once, don't watch it twice. Watch it about 75 times. Put it on a different computer screen and make sure you can follow along with that slide in your notes that has it written out plain. Explain it to a friend, explain it to your mom, explain it to your grandma, explain it to your dad. Make sure that you can explain how in the heck does cellular respiration actually produce the energy necessary for life. Teach your family and friends how saying mitochondria is the powerhouse of the cell is a bit of a gross oversimplification of the process. Because holy smokes, I hope your brain hurts at this point. It's so stinking complicated and beautiful. Go make sure people in your life know what's going on. Try this practice question here. It's a really good one to practice for yourself. What would happen if that membrane was permeable? What would happen if the ion H plus could just travel through the phospholipid membrane like it wanted to? Here's the oxygen lacking thing that I told you I would talk about. So sometimes you don't have oxygen. I like to think of this mostly like a sprinter. There's other uses, we'll get to those. If you are a sprinter or you are sprinting from one side of the field to the other, I'm talking a full out suicide top speed, let's go. You're going to run out of oxygen. It's just a guarantee that you will run out of oxygen. What happens at that point is your body continues to do its best to convert glucose into energy. It's just that it gets a little stuck. And so it can't quite go all the way. So a couple different things are going to happen. And you can see two of them here in A and B. Sometimes you don't have oxygen that's necessary. Sometimes you live in such an environment that oxygen is not an option for you. You don't have any. Or you're just an organism that doesn't have a mitochondria, so you can't go through this process. If that is you, you are known likely as a little organism known as yeast. Think about where you have ever heard the word yeast used before. Think, think, think. Hopefully you came up with something like, is she talking about bread? Yes, I am. I'm talking about any and all yeasty baked goods that you would make. There are tons of them that the base product, the thing you start with, is yeast. Yep, that's the one that works here. It doesn't have mitochondria, and it is pretty freaking awesome that it doesn't have mitochondria because it does something else instead that is delicious. So again, if it's no mitochondria, it's only cytoplasm in this case. And what happens is you don't get energy per se. You get this stuff called lactic acid. That's the burn, if you've ever felt the burn from sprinting. Or you make this cool thing called alcohol. So here's how the alcohol part works. Alcohol is thanks to yeast. If you know anyone who does any brewing or is in brewing sciences, it's really a whole study that people do now. They feed yeast and sugar together. When you put yeast and sugar together, they go through this process and they end up making alcohol. We were talking about the things like beer and wine and spirits and all of those things that I know that you are responsible and you will wait till you are at a point in your life where you can consume them responsibly. Know that it would be my honor if you said, ooh, I remember this is just cellular respiration without mitochondria. And please make sure that you're around biology friends and not other people, because then they laugh at you. But it's okay. I'm proud of you. The second thing that happens if you have a mitochondria, but you do not have the access to the oxygen, so you can't go to the green, is you stay on the blue side, and then you can make that lactic acid. That's the burn that happens in there. So if you've ever felt the burn from sprinting really, really fast, you don't get to do that. Aerobic process. If you've also heard of things being aerobic and anaerobic, remember an as a prefix to anything just means without. So it just means without energy, oxygen, you don't get to make the ATP, but you get some other cool stuff that's pretty freaking awesome. So just some differences there, a little sports connection, a little connection to later in your life when you will be responsible and be doing this. We are going to do a lab that has to do with fermentation, but we're going to do it safely and responsibly. And I will talk about that with you if you're watching this early, or give it a little bit and then I will talk about that. It is known as your bread lab. That's right. We're going to go play with fermentation. We're going to play safely with it in just some bread. I'm going to have you do some baking and we're going to just see what happens. I'm going to let you play with this process because it's pretty yummy to play with. Please go take your formatives. If you have any questions after taking the formative, if you want more information, if you want more practice, my best hint and tip in this case is flashcards, and then write out the process and then do it again. And then close your eyes, go to sleep, wait 24 hours, try it again. Close your eyes, go to sleep, wait 24 hours, do it again and again and again and again and again. And eventually, I promise, one day it'll just be stuck in your head. And then you'll know everything that you need to know about cellular respiration. Let me know what questions you have.